Welcome to BioBox Labs. Today we're going to talk about photosynthesis. Did you know that the sun shines light on plants and they're able to capture the energy from the sun and use it to convert carbon dioxide and water into usable energy for them to use for all of the cellular activities that take place in plants. We also get to use some of that energy when we eat a plant or when other organisms eat plants. So all of us ultimately get our energy from the sun through photosynthesis. So did you know that photosynthesis also produces another product, and that is something that we use every minute of every day, and that is oxygen. So today's experiment, we're going to see how photosynthesis produces oxygen, and we're going to use a plant called Elodea. Come along and let's show you what we're going to do. For this experiment, all the supplies that you need have been supplied in your BioBox. But if you want to join us and are not part of BioBox Labs, you may collect a couple of these supplies. We're going to start with Elodea, which is an aquatic plant, but you can use other aquatic plants that you might find at a pet store. We have forceps. But you could even use any kind of tweezers. We have a funnel. This one happens to be 65 millimeters. 250 millimeter beaker, a 20 milliliter test tube, a ruler, and a big bowl filled with water. So what we're going to do is that the Elodea is going to be our plant that's undergoing photosynthesis. So in that plant are all the leaves with chlorophyll that are going to capture the energy from the sun. So first we're going to just rinse it off to make sure it's nice and clean. And then we will start preparing our apparatus. So you're going to have a beaker, we have our Elodea, and we have this funnel. So first you're going to curve the Elodea into a circle and stick it inside the cone of your funnel. And then you're going to set this, hold that circle in there, into the beaker. And it's okay if the Elodea kind of falls down like this. And if you have the tip of the funnel where the beaker spout is, it will help add a little bit of extra room. And then ultimately what we're going to do is we're going to set this up underwater, but we're going to end up putting this test tube so it fits right over the top of this funnel. So this is what we're going to do, but we're going to do it underwater. So we want to capture water inside of it. That's why we have our big bowl. So as you set this all up, put that together, and now we're going to put the whole thing underwater so it completely fills up, and you'll see it bubbling away. And then we're going to take our test tube and also kind of tilt it up so that it completely fills with water. Then we're going to put them together like I showed you before, where we're going to put the test tube over the tip of the funnel, and now we're going to angle them up, and amazingly the water stays inside of this test tube, and you want it completely filled because the water is important for photosynthesis, but there's also carbon dioxide dissolved in the water so that the plant has everything it needs for photosynthesis. Now we're going to set it near a place of sunlight. So you either want to put it in a really bright window, or you can put it under a very bright lamp, or even better, put it outside in the bright sun, and you're going to begin to see oxygen form. Now we're going to let it sit for 2 to 3 days to let photosynthesis occur. Remember, if it's just inside and the light is not real bright, it probably needs more time. If it's in this hot sun outside, it will probably produce more oxygen more quickly. So let's check back. So it's now been 2 days, and we can see that photosynthesis occurred because there's a tiny oxygen bubble on the top of our test tube. You can't leave it in bright sun too long or the Elodea will start to go bad. But if you've let it in front of a light, maybe you need a couple extra days to get any amount of oxygen coming to the top. Now let's take a close-up look at the oxygen that's been produced. Take a close look. You can see down here at the bottom, there's even little bubbles along the funnel, and there's a larger bubble up here on the top of the test tube. If you tap even on the test tube, you can even see that some of the little bubbles will rise to the top. Now maybe I've tapped it now so many times. There we go, there's a couple. They will rise up to the top. But now we can measure the air bubble to see how much, and we'll measure it in millimeters. So if I set this here, we're going to measure from the water level there up to the top of the test tube. Oxygen bubble is about 5 millimeters, 5 little tiny lines on there. So 5 millimeters of oxygen have been produced over 2 days. Now, yours might be more or less depending upon the amount of oxygen or the amount of sunlight that it was given, but each one of these, the Elodea, is going to produce some oxygen. So now to make it really interesting, we're going to see if light really makes a difference in photosynthesis. So for the next 2 days, you're going to take your same apparatus, stick it in a closet, and see if the oxygen bubble grows at all. After 2 days, check it out. Thanks so much for joining us on our scientific adventure as we talked about photosynthesis and how we learned that oxygen is an important product of photosynthesis. If you enjoyed this, join us on bioboxlabs.com and see what other adventures we are up to. Thanks for joining us. We'll see you next time.